Hi, my name is Mark, and today I will be presenting our work on a single-cell data visualization tool called VITESSE, which we are developing in the Gellenborg lab at Harvard Medical School. There are now many labs generating datasets at single-cell resolution, which span multiple modalities, including transcriptomics, chromatin accessibility, and imaging. After cells have been profiled and the raw measurements obtained, standard processing steps generate further derived data, which may include dimensionality reductions, cell segmentations, and unsupervised clusterings or cell type assignments. While there are currently many tools for visualization of individual single-cell modalities, such as genome browsers, image viewers, and scatterplot viewers, there are currently few tools which enable visualization of all of these modalities simultaneously. In this plot, taken from the list of published datasets from the NIH Human Biomolecular Atlas, or HuBMAP consortium, we can see that HuBMAP labs have already submitted hundreds of datasets across technologies and organs. We would like to provide web-based interactive visualizations for all of these datasets, which requires rethinking existing visualization approaches in terms of both human-computer interaction and software engineering. To address these challenges, we have developed a framework for interactive visualization of single-cell data called VITESSE. VITESSE is modular and can be configured to display different visualizations depending on the data produced by a particular experiment or technology. In this screenshot on the left, we see a dataset containing spatially resolved measurements and cell segmentations, visualized in a spatial view, alongside scatterplots for multiple dimensionality reduction results, a heat map to show the cell by gene expression matrix, and statistical plots such as violin plots and histograms. VITESSE loads data from static files and does not require a specialized server, reducing infrastructure costs and making it easy to deploy. To enable relation of entities such as cells, cell types, and genes across plots, VITESSE includes a flexible coordination model to enable plots to be linked on any subset of properties. In addition to the web application, VITESSE can be used in Python and R environments. VITESSE can load data from data objects, simplifying the data conversion process. VITESSE can be used not only as a standalone web application, but also as a component embedded in another website or an interactive widget in Jupyter Notebooks or RStudio. With its modular architecture, VITESSE can be used as a viewer for microscopy data, RNA-seq, codex, and ATAC-seq, among many other modalities. VITESSE is currently being used in the data portal for the NIH HuBMAP consortium, where it is embedded to visualize HuBMAP datasets that have been processed uniformly. I'll walk through how the features of VITESSE support each of these use cases. This is a light sheet microscopy dataset, which showcases the volumetric rendering features included in VITESSE. The image first loads as a 2D slice, but we can use the drop-down to select a 3D rendering mode. The data for the volume takes a little while to load. It is loaded from an OME-TIFF file stored in a cloud object storage system. Once the data loads, we can pan and zoom around the volume. We can use the volume rendering controls to select a maximum intensity projection mode. VITESSE also includes controls for clipping along the X, Y, and Z axes. We can use the Z slider to focus on a smaller section of the volume. We can use the image channel controls to select a different color map or change color scale ranges. For single-cell ATAC-seq data, we can use VITESSE to visualize chromatin accessibility on genome browser tracks. As we zoom in along the genome, the genomic data will load at higher resolution, and we will see additional gene annotations. We can coordinate the genomic visualization with the scatter plot to highlight individual clusters across both plots. We can change the rendering properties as well. This is a single-cell RNA-seq dataset for the lymph node. We can select a gene such as CD79A, which we will see is highly expressed in 2 clusters, which contain B cells. The violin plot updates when a gene is selected to visualize gene expression distributions for each cluster. This is an example of a codex dataset, which contains imaging data, a t-SNE dimensionality reduction, and a cell by antigen heat map. On top of the image, there is a cell segmentation bit mask. The test supports multi-scale bit masks, which render in higher resolution upon zooming. We can select an antigen, and the cell segmentation mask colors will be mapped to the antigen quantification. The scatter plot here contains over 70,000 cells. We have implemented a dynamic point opacity feature which prevents overplotting and makes it easier to see the structure in dense clusters. In this example, we see the test embedded in the HubMap portal on the details page for an autofluorescence microscopy dataset. The test loads the image data directly from the OME TIFF file, in this case with 3 channels, and we can use the image layer controller to dynamically change the color mapping or add channels to the visualization. The test supports pyramidal image files, so as we zoom in, higher resolution data is loaded, and we see the image become more detailed. We are currently working on improving the test to make it easier to use and to support additional use cases. We are working to better support multimodal datasets stored in standard multimodal data structures. Next, we are working to support large MERFISH datasets containing tens of millions of transcripts and hundreds of fields of view. We are also working to integrate the test with statistical and machine learning methods. Specifically, we are adding support for the MU-data format, which is a container around multiple AND data objects, so that we can point to a single data structure on a cloud object storage system to load a multimodal dataset. We are making performance optimizations in the spatial and imaging view to scale to larger datasets. And finally, we are working to build plots for comparative visualization of multiple datasets to support reference-based analysis tasks. To create your own visualizations with the test, the Python and R packages are great starting points when the data is in a compatible data structure. This notebook demonstrates the usage of the test Python package. We can first import the functions from the package, then we can load the data into an AND data object. We create a Vitesse configuration by specifying the data and plots we would like to include and configure their arrangement in a grid. In the final cell, we render the widget. We see the 2 scatter plots on the left and the heat map on the right. We can interact with the widget, for example, selecting a gene. In Python, the package currently supports loading OME TIFF images, as well as AND data, Loom, and Snap Tools objects. The R package currently supports loading OME TIFF images, as well as Seurat, single-cell experiment, and Giotto objects. We are working on supporting additional common single-cell file formats. The Python and R packages also contain functions for exporting data to cloud object storage systems. Once data is in the cloud, a new configuration can be defined based on the remote files. This configuration object can be converted to a URL to save or share with collaborators. The test contains custom visualization components, but also leverages existing open source visualization libraries, including HiGlass for genome browser tracks, Viv for loading and rendering 2D and 3D imaging data, and Vega Lite for statistical plots such as bar charts and violin. To learn more about Vitesse, you can visit vitesse.io, where you will find installation instructions, tutorials, example notebooks, documentation, and details about our future plans. I'd like to thank the other members of the Gellenborg Lab and acknowledge the funding that makes this project possible. Thank you for listening to my talk, and please feel free to send questions or comments through the conference website.